Hi everyone, and welcome to our 5th daily booster. You'll notice that some of the content in today's booster is listed on our advanced information list. So the first thing we're going to look at today then is the idea of metabolic rate. Now, when we're talking about metabolic rate, this is the speed at which your body is using energy. And different people have different metabolic rates. So what we find is that certain factors like your age, your lifestyle, so whether you're really active or quite sedentary, so you spend your life sitting on a sofa, and also your gender, play a role in how much energy you are going to use and therefore how much food you need to eat. Because what we find is the higher your metabolic rate, the more food you need to eat to provide that energy. What we're now going to look at are the different types of food substances. Now, as far as our pre-release information goes, then this is where it's coming in for the practical activity. So it's practical activity group 2, and this is listed for the foundation students and higher students. So we need to know how to test for these different biological molecules. Before we can delve into that, we need to understand what they are, because if they're asking us to know how to test for them, it wouldn't surprise me in the slightest if there was a question linked into that they haven't explicitly stated on there about carbohydrates, proteins, etc. So carbohydrates are first. Carbohydrates are polymers which are made from the monomer of simple sugars. By that we mean we've joined lots of these sugar molecules together to make a long chain, which is our polymer, the carbohydrate. We use carbohydrates for energy. They're one of our major sources of energy, and we will break them down using an enzyme called a carbohydrase enzyme. And remember, we can always spot an enzyme because it ends in ASE. Now, what we actually have is a good example there, starch being broken down by amylase. Now, amylase is present in your saliva, and that's why if you have bread in your mouth for a reasonable period of time, then you start to notice it gets a sweeter taste, because the amylase in your saliva is breaking the starch down into those sugars, and you taste them. So there are two tests that we can do here. The first one, testing for our reducing sugars, we use a substance called Benedict's reagent. Now what we do is we add the Benedict's reagent to our solution with our food, and we're going to need to heat it in a water bath. So they could potentially ask us about changing water baths. So if you're using a Bunsen burner, for example, as a water bath, how could we improve it? Use the electric water bath. Now the colour change we expect to see if reducing sugars are present, it will go from blue to red. We then also have the ability to test for starch, which uses iodine. And all we do is we add the iodine, which is an orange-brown colour, and if starch is present, it goes blue-black. The second biological molecule we're going to look at are proteins. Proteins are again polymers, and they're made from amino acids, which are the monomers. So what we actually find is that the sequence of amino acids is going to determine what final protein we have. And we've already had a little look at how we make those proteins in one of our earlier boosters. Proteins are used for growth and repair, and they're broken down by protease enzymes. So the biological test for our proteins is we use this stuff called biuret, and it's going to change from blue to a purple colour. The third biological molecule are the lipids, also known as the fats if we're going for simplistic terminology. Now, lipids are very important to organisms because we use them for energy, first of all, but also for insulation and buoyancy. So buoyancy particularly important if we're talking about aquatic creatures, so the ones that live in water. In terms of what lipids are, they are not a polymer. So this is the one that's not a polymer. They're made from 3 fatty acids and a glycerol molecule. So what you actually end up with is you've got your little glycerol molecule here, and then we have our 3 fatty acids that join onto it. So that is what our lipid looks like. Glycerol plus 3 fatty acids. In order to break them down, we use lipase enzymes, and because they have those 3 fatty acids, when we break them down, we are going to see a decrease in pH because we're releasing acids, as the name fatty acid suggests. So breaking down lipids leads to a decrease in pH because we're releasing fatty acids. To test for our lipids, quite simply, we add some ethanol to our solution, give it a bit of a shake, and then if lipids are present, we get this milky white emulsion in the tube. Now, that leads us very nicely on to the idea of respiration, and we're going to look at both types of respiration in our booster today. The first one is aerobic respiration. Now, aerobic respiration is the one that's going to happen when we have plenty of oxygen available. This is the respiration of choice, if you like, for organisms, because we get a lot of energy from it. And this is how we're going to provide ATP to all of those living cells. So respiration happens all the time because we need it to generate that ATP. It is an exothermic reaction, which means it's going to give out energy, so it warms things up. And it's going to take place inside that subcellular structure of the mitochondria. One thing to note here is that when we're thinking about different cells, they will have different numbers of mitochondria present based on their energy requirements. So if you've got cells that need lots of energy, then they're going to have lots of mitochondria. So things like muscle cells and liver cells, loads of mitochondria because they need loads of energy. Your skin cells, only a very small amount of mitochondria because they don't really need much energy there. What we find is at GCSE level, we're going to say that aerobic respiration is glucose plus oxygen making carbon dioxide and water. If you go on to do A-level biology, this is going to be a vast oversimplification for you, so prepare yourselves mentally for that. But GCSE level, that's all we need to know. Make sure you do know how to write the balanced symbol equation as well. So glucose, C6H12O6, and then we're going to add oxygen, which is O2, and we've got 6 moles of oxygen, and that's going to make 6 moles of carbon dioxide and 6 moles of water. So you should know O2, CO2, H2O, they're standard formulas you should know. And then obviously glucose, C6H12O6. Just remember the balancing for this is 6 is in front of everything but glucose. You'll notice at the bottom I've given you the actual numbers. So 38 ATP molecules are produced in aerobic respiration. And you can see compared to anaerobic, 2, it is much more in terms of the amount of ATP we make. Hence why this is the one we go to first of all. That's not to say anaerobic respiration is irrelevant, because there are going to be times when we don't have enough oxygen to carry out aerobic respiration. So we're going to swap over to anaerobic. Now, anaerobic respiration is just going to convert our glucose into a new substance, lactic acid. So we still make a little bit of ATP, those 2 molecules, but it's not a lot. Now, the whole purpose about this is it allows us to transfer energy when oxygen isn't available. So this might be in examples where the soil has become waterlogged. You can't just not make ATP in those situations. This gives us a backup plan, if you like. It is worth remembering that in these situations where we have created lactic acid, we are going to need to break it down because it is toxic. So what we actually find is that we have something called oxygen debt. If we think about humans and exercise, when you've been doing a lot of exercise, you actually then slip into anaerobic respiration. So you make lactic acid, and that's why your muscles start to feel fatigued and you get those kind of aches. What we need to do to get rid of that lactic acid that's built up is we need to then keep taking in oxygen, which will oxidize the lactic acid. So that's what we mean by oxygen debt, is we need to keep taking in oxygen post-exercise to break down the lactic acid. Another type of an anaerobic respiration is fermentation. We will touch on this again in Unit 6 of Chemistry, but fermentation is anaerobic respiration. So we take glucose and then it's going to be changed into ethanol, which is an alcohol, and carbon dioxide. And again, the balanced symbol equation is at the bottom there. That's not to say that you must memorise that whole thing for your biology paper, because if you've actually done some work on your chemistry and you're doing triple, then you know all about the formulas for alcohols. So you can work that out in the exam if you've got that knowledge. Alternatively, you can just memorise it. As always, what we need to now do is go and double check our knowledge from today's little booster. So head on over to the quiz. Again, I've posted the link in the description box below. Don't forget to join us again tomorrow for our next booster.